Hello.
Shriram, no problem.
Yeah,
Okay,
yeah.
so shall we start now?
Yes, madam, sure.
Okay, so I will share my screen.
By the way, ma'am, happy Ramzan.
Okay, thanks.
Yeah.
Okay, so have you tried earlier executing this star chip or working with it?
No, ma'am, actually, I tried installing the packages, but yeah, I tried even setting my the Perl path according to my Perl path in the workstation, but still, I don't know why I'm not able to execute that. Though
All
I
right.
tried to run it superficially just by seeing the GitHub, and I got to know like how it works to some extent. So I thought if someone can help me out.
Okay, sure. So let's start with the. Let me share my screen first. Then I can. Just a minute. I'm not able to share this. Yeah. So is it my screen visible there?
Yes, yes.
Okay. Okay, that's fine. So, mm, okay. So we'll start with start shift first, but for before that, we need to know that on what type of data it works actually. So you must have already gone through the paper which is related to star chip, right? So they have mentioned that it works for back-spice junctions to detect the back-spice junctions within your RNA sequence that we are
Yes.
having. And to identify or to detect backsplice, where exactly backsplicing is happening in your sequence, for that we will be using STAR tool for alignment.
Yeah, yeah.
So, so let's first go through theory so that we can understand the.
Your voice is actually breaking and coming back.
Okay, is it not clear?
Yes, madam.
Okay. Uh, okay. So when since the tool name itself is star shape, so this is for chimeric sequences that we are identifying. And why star? Because we will be using star alignment tool for it. No matter there are other alignment tools available, but for
Yeah,
this
yeah,
specific tool, star will be included for alignment.
yeah,
So you can use it for like two ways, whether you are running star before starting analysis with the star chip or even included within that. So
yeah,
those instructions are already given in the manual,
yeah,
and it helps in identifying or addressing the challenges of high false positives rate that were earlier identified using like if you are using other alignment tools. So for that it has specified that when you are using star, you will be able to overcome with those challenges. And also
mm,
if we have long reads from sequencing platforms like PacBio or Nanopore, so those type of tools also you can use.
mm.
Yes. So just a minute. Yes. So what we can do? These challenges we can overcome because usually we are dealing with the data from Illumina platform. But even if we have long reads, then also we can use Starship with
Yeah.
our with the Star to perform the analysis. So theoretically, first we will go through, then later we can go with the practical session. But still, I need to inform you that. In practical session because these star alignment and all take a long time, so
Yeah, I
that's
know
why.
STR, ma'am, actually how to
Yeah,
run. You just,
yeah.
I don't need it from scratch. Like wherever I feel doubt, I will ask you,
Yeah.
madam. Just
Okay,
explain.
okay, sure. Okay, sure. So we know that Starship is written in Perl, Bash, and R. So these tools will be need to be installed when you are running Starship. So these are like software dependencies for Starship. So
Yeah.
before running that, we will be using them, and then we have within Star when you are aligning your samples, you have specific parameters that needs to be there while you are running to get the like chimeric alignments or positions within your reads when you're aligning the data. So we'll see how to
Can you
customize
explain
it.
that parameter actually? Like generally, so segment minimum means the overall length of the chimeric rate should be 15, and the second one is like on either side of the fusion junction, the minimum length should be 15 so that it can be considered as a chimeric rate, right?
Yes, this is the minimum length. Even if there is an extension or gap between the two sequences, nucleotide set of sequences, more than 15 nucleotide bases, that also will be detected, but minimum will be 15. That we are. So these are not default parameters
Yeah.
for your star usually then we when we do alignment, but when you are going for star chip, so you need to be having these parameters, and there are other parameters as well, so that we'll see in some time. And yeah, okay, so let's see.
Can you again once in short explain that two parameters? I just got confused.
Okay, so there are okay. These are specific parameters that we are, so the specific parameters that needs to be there, but there are other parameters as well that we will see. But right now here we what it is mentioning that identify the chimeric segments that have minimum length of 15.
Okay.
15 nucleotide sequences. Okay,
Okay.
so when if it is more, then it will also be detected, but less than 15 will not be detected, because in that case it is not sure that it is making a complete circular RNA or not, because sometimes nucleotide pairs which are mapping is just 3 nucleotide long, so that in that case we are not sure, right, that it is making circular RNA or not. So that's why we are keeping the parameters. We can also modify, but these parameters are standard parameters for Starship to
Okay.
run on your data. And then when chimeric junctions are there, when for example the sequence nucleotide sequence we keep ATGC, and then after some unmapped reads there are other set of reads which are mapping, so there will be a gap in the alignment, right? So one some part of one exon is mapping to other part of some part of other exon, so exon 1 and exon 2, or even exon 1 and exon 3. So because chimeric reads will be non-contiguous, so it is
Yeah,
not
yeah,
necessary that exon 1 and exon 2 reads are being read for
yeah.
splicing junctions. So that's why when the overhang is minimum 15 sequences, 15 nucleotide sequences are is there, for example the gap or the reads between them should be minimum. So because this segment that we are already keeping 15, the first parameter, it is following the second parameter itself. So this like these two are similar, but specifically specifying that overhang minimum should be 15. Okay, so this is follow up a parameter for the first one where we are mentioning the segment minimum, segment length. And okay, just a minute. Yeah, so we'll see other parameters also in star when we are running star, so we'll check in some time. Okay, so here, as we have seen in the paper itself, that what type of alignments it is able to read, that
Yeah.
if there are like there is 5 prime reads, and then there are 3 prime reads which are, and some sequences are mapping in this way, so we can see, we can this star chip will be able to identify these type of sequences as a circular RNAs.
Yeah.
And okay, so this is how it follows, that when back splicing reads are there, so it will try to first, when we are aligning our reads, first it will try to map to the reference genome and see that whether the some of the reads are from 5 prime end, and some reads are from 3 prime ends, and those are mapping in particular sequence, so that it can be declared as circular RNA. Those will be detected and identified by your tool, and then the matrix will be generated to inform that how many number of genes are there of this type where circular RNAs are present. And further we can annotate based on the genes and what exons exactly it follows or it falls into, and based on the strengths.
Can you
So
again go slow, ma'am? Can you go slowly, like how this actually works? Like first part is what to do with the FASTQ file.
okay, so first we see that there are reads that that are from five. There are reads that some part of reads are mapping to the forward strand, and then some part of the reads are mapping to the reverse strand. When we are making, for example, ATGC is following on the first segment of the read, and it is mapping to your genome, and the second segment is ATGC again, which is also mapping to the other part of your genome. So
Hmm.
it will try to identify these type of sequences which are also forming a combination to make a circular RNA. For example, here as we see that this particular read, when we uh, merge these two reads, which are half part of the first read and half part of second read, so it will be aligning in such a form to make a circular RNA. So
Yeah.
this type of reads will be detected from your FASTQ files when it
Yeah.
is aligned to your reference genome.
Yeah.
Because when we are aligning to the reference genome, then only we will be able to know the order of the sequences which
Yeah.
are there. And then after that it will try to like quantify how many number of reads are there in this form, and what are their order, what are their specific chromosomal location, and all. For that we will be doing indexing for our reads in our FASTQ file, so that we can know the proper order of your reads in your whole genome, that it is lying on chromosome 1 or in which exon sections. And based on those annotation that is performed by the alignment and the indexing phase, based on that it will try to identify and quantify the number of circular RNA or that type of reads which are identified through your through your star chip. Okay, so that's how it works. So when we go step by step in the flow chart, then you will be able to clearly
Okay.
understand that what is exactly happening. Okay, because there we have specific instructions. So let's just move
Yes.
to the script part maybe. So when we see the workflow, so we need to first process the sequence that we have with the star. So where we are doing indexing first, and then so that it will be, the reads will be in exact order how our genome is, based on the chromosomal orders. And then after that, after alignment, it will try to identify the chimeric reads, the type
Mm.
of reads which are non-contiguous in your sequence that you are providing as an input file, the FASTQ file. After that, the chimeric reads which are having probability of making circular RNAs will be filtered and annotated based on that. So for that, you for different part of workflow, we will need different inputs at
Yes.
each stage, and we will be generating these input files like chimeric junction files and the file, reference genome files from different tools, and also we will be downloading from websites. Okay, so and then we have the requirements for running the Starship, because if we are running star alone,
Yeah,
we
I
don't.
have the requirements, all the necessary requirements for that.
Sorry.
So yes, yeah, all the software dependencies, whatever I need, I have actually.
Okay, so yeah, if you don't have, you need to install, else you can run directly. Okay, so to run Starship in your on your data, you need to clone the repository that is there in the GitHub. Okay, so for that we need to just give git command, and then you can add the link for that repository,
Yeah, yeah,
clone the repository. Okay, and once you are adding that, you can make your Perl script that will be used for Starship analysis
yeah,
for executable. So for that, you need to change the mode of, like, give the permission for your files to be accessible. So the Perl script that you are, it should be accessible by your command so that it can run, and the dependencies you know already and have installed. So this is maybe you are mentioning that you are not able to run the Perl script, so
yeah,
that could be the permission issue. You need to exactly check in the error when you are running the program, that what error is trying to say.
yeah, madam. Actually, I tried even changing the Perl path than the one that is actually present in the main script. But still,
Mhm.
I don't know where going wrong. You better explain, madam, so that I will know.
Okay, so yeah, we will see the file orders so that you can see where the files are, and where the script is, where your file needs to be there. Okay, so first we are doing the genome indexing. Once we have the genome file, which is the chromosome FASTA file and the GTF file that you need to download from database,
Yeah, yeah,
whichever database that you are using, and then once you run the genome indexing file, then your indexes will be generated
yeah,
for your genome. Okay, so that will be like proper order of your chromosome and locations, that chromosomal locations which are there, and chromosome
yeah.
length files is there from where your chromosome is starting.
Index of a book, we can say.
Yes.
Yeah,
And then we will move for alignment. So this alignment step is optional here, because we can do the alignment within Starship itself, but we can prefer it separately, because when we are running a Starship with a star, so when there is an error, it will be difficult to understand that whether the error is in alignment step, or even for quantifying the circular RNAs. So better to run alignment fast and then go for circular RNA quantification with
yeah.
starch. Okay, so here we have different parameters where
Mm.
all are not
Huh.
okay, all of these are not default parameters. For them, we need to see that what parameters that we are keeping here, so
Yeah,
that we can run the star in such a way so that it will be identifying the junctions or the splice junctions that is there within our data or
yeah.
within our sample. Okay, so here we have in the end, we have some parameters. First, we are mentioning somewhere that there should be match of minimum one nucleotide, and then there should be mismatch if there is a mismatch of two nucleotides. So this way we can modify it always depending on the data. For example, if we have the sequences, like sequence length is very less in our data, for example 10 base pairs
Yeah,
reads are there, so for that we will be modifying. For example, if it is 10 only, so we cannot make it minimum 15, right? Then in that case it will not be identifying any circular RNAs
yeah, yeah,
on junction files. So based on the data also we need to that's why data pre-processing is important before processing,
yeah, yeah,
because we need to know the data, and based on that we will be modifying these parameters, and then we will be getting the junction file, which is the chimeric junction file in the last command that I am mentioning, and then we need the SAM file,
yeah,
so the same junctions in the SAM file format.
yeah, yeah.
So both will be required by the start chip, and
Ma'am,
then you can get, yes,
ma'am, I have a doubt. This
yes,
separate SAM fold, when I
yes.
saw the manual of star, it said that this command is that is actually not working, or it is somewhat masked actually. Star manual is saying. And I tried running this, but I'm using the chimeric out junction part actually. So Sam, it's saying it will be decaparated, or it's decaparated like that.
Yeah, so they are mentioning because it is SAM is usually not required when we are running a normal alignment,
Hmm.
because in that case we need the sorted BAM files,
Yeah,
right?
yeah,
But if we are mentioning the to get the output in SAM file as well, because it is required by our star chip, so we need to strictly mention in the here that we need SAM file as well.
yeah,
Okay, so in that case, bed file will not, BAM file will not be generated, but we will get the SAM file.
yeah,
Okay, so we can use it when it is required.
yeah,
Why star it is mentioning? Because it is not usually required. That is an additional file size that is occupied on your disk, right?
yeah,
And
yeah,
is not separately required because BAM we are already getting without this input,
yeah,
and
yeah,
BAM we can visualize in IGV or other visualization tool where it is required. SAM
yeah,
is usually not required, just a text format of file, and that is of very large size because it is in
yeah,
text
yeah,
format. So that's why it is not required, but we can use depending on the objective. Now we want
yeah,
to run a star chip, so we need it. That's why we need the junction file, chimeric junction file, which is chimeric out dot junction. Yeah, we need the chimeric outdoor junction file and also the chimeric out.sam file. So both the files we will be getting. Additionally, the other files, alignment files that we having the about the information about splice junctions. So that we will get with the star, and these files we will be using for star chip. Okay,
What is
so
the difference between these two files, dot junction and SAM file?
yes. So here the junction files will be having the information about the all the chimeric alignments, that what is the chromosomal location of your alignments that is read by there. But in SAM file we will be having the data in tabular format. So
Yeah,
that's why this is for our visual understanding, that we can open in any text format or in Excel, and we can see that how the data exactly looks. And
yeah,
and that is also used by Starchip. So it supports tabular format input. That's why we need the SAM file.
yeah.
Okay. And then so here we I have added the default values that starting. If you are not mentioning the specific values here with the align junction, overhang minimum, and the other values that we are adding, so star will take the, even if you are skipping these parameters within a star and just you are giving a star and the thread information, without thread also you can run, and just the input reference directory and the sample information, then also star will run. But in that case, you will not get the junction file and the sam file, and also your parameters will be set at the default values that is
Yeah.
given here. Okay, so because why we are keeping the parameters as per our requirement, because we know our data, we know the length of the reads,
Yeah.
read length and all, so that's why we need to customize based on our requirement. So yeah, so based on that we can modify always of our data. And then once to run the star chip, once we run the starship command to clone the directory, we will get to the folder, starship folder, with all the these folders within it, and the script, that is the Perl script that it has, and the bash script to for the setup, starting setup of your start chip. Okay, so this start setup script we will be using to generate the bed file for our samples to run further for the start chip. Here we are just cloning the directory so that we can get all the necessary details within our start chip folder. And yeah, so Starchip is because used for both circular RNAs and fusion detection. So that's why we have both the scripts here. So as
Yeah.
per our requirement, we can run that. So here we are using the Starchip setup.shell script to get the bed files that is required by Starchip. Okay, and we will be getting the for our reference genome, or the data that we're having for the reference genome, we will be using that to convert that in the into bed file, because that is required by starchip script, Perl script, to generate the results.
Yes.
And then we need to customize the parameter file for Starship. And in the Starship folder itself, it is given the it is there is a example parameter. Okay, is my screen shared?
Yes.
Is it there? Okay. Okay, just a minute. I think I don't know why it stopped. Just a minute. Here, let me just show you where is Starship. Can you see the files here, the folders? Is it visible?
Yes, ma'am.
Let me just make it. Um, okay, so here we have example parameter files that we can see to generate, because it is recommended by the Starship manual itself that we need to generate exactly same parameter file
Yeah.
that is having the read cutoff and all the values that it has. Here is the Starship for circles. So we need to open that file for us. So is it visible for you? You can see here the for parameter for Starship circles is given, where reads cutoff is 5 and minimum subject limit is 1, number of CPUs that it will be using for analysis. Then do splice equals to true, that whether you want to include the script to read the splice junctions or splice reads. So that
Yeah.
is true. Then CPM cutoff value equals to 0, and then again cutoff value is 0. So this is the default parameters that is given within an example parameter file that you need to have. And then again, because we are concerned about the splice junction, so we need to keep it
Mhm.
true. And then annotation true, these are default, it is given. But what you need to change is here the reference bed file. So you will be changing the here path of your reference bed file here, that which is GTF exons files that we just saw the in the bed file generation. And then reference FASTA file, so where is the your reference genome file, this path you need to change.
Yeah.
And then okay, and then you have star parameters. If you want to run star within star chip command, then you can keep it yes or no. But here first we need to give prefix. If you have any prefix for your star output files, that I will show you in minute. If there is no prefix in star results, then you can give it just blank.
Yeah.
And then run star if you want to run, so true or false. So we can give it false because we are already separately running star, so we can give it false. And then we need to give the path for where our star results are there. Okay, so this star prefix is for the results that is generated by your star alignment step. And if your files have any prefix, for example, if you are running and giving project ID for your all the results, so that you can add, or else you can keep it blank. Most of the time we keep it blank itself with the command that we are using.
Yeah.
And then if you have the, this is again about the
Yes,
star
madam.
file, that zcat for fastq files that you are having. But since we are not running, so we will try to hash it or comment it out, so that when we are running the star chip, it will not include these particular commands from this file, because we are not using star. We just need to mention run star equals to false, and other things we will keep it in the comment lines. And then this id step back is required. If you are have you for you are running only one sample, then you can give it one. But if there are more than one samples, and then you need to keep your star results in different folders with sample names. And accordingly, for the sample two, if you have two samples, then you can give it two. And accordingly you can give this id. Okay, here example is mentioned here, that how you need to give your ID here as a step back. Okay, so that you can check. And accordingly we can modify. But always remember, if you are running a star, just comment these lines, otherwise it will throw error.
Yeah, madam. So if you show the examples, then I can understand. Now
Okay.
it's making like if you do, I mean, if you do practicals, practical
Yes.
session, then I will get to understand.
Yes. Okay. So yeah, so that's what we have mentioned here. Let me. Okay, I will show you the exact um, script that we did. And then once we have generated the parameter file, and then the other file that we need is star directory file, where we will be adding the path of your star results that we have generated in during star analysis or the alignment step. So these two files we need. And then after that we will be using those two files to run the star chip circles.perl script for based on these two files. So first file is having the path of your star results, and then second file is the parameters file where it is running. And the output we will get in matrix, and then matrix and images format, where we will be getting the pca plot and all the annotation of your circular rnas that is identified by your star chip. And once we are running the starship.circles Perl script, then we will be getting two bash script, which is step
Yeah.
one and step two. So this is generated itself when we are running the starship circles Perl. And after that, once it is completely successful, the Perl script running, then we can run the step one and step two shell scripts one after other to get the further results. So for step one, what it will do is it will filter out the chimeric reads that appear circular, and then so these are the log that is generated when we are running the step one shell script, and then filter circular reads based on the assigned thresholds of 5 reads in 10 individuals. So that is for if we are using 10 samples or number of samples, based on the number of samples. So it takes by default 10, but you can add more as well. And then it will generate circular RNA FASTA and GTF files for realignment and quantifications. Then the quantification will happen in the step two, where your Lima package and HR package is required. So we can install with using
Yeah.
bioconductor, or even if it is available in CRAN R repository. And then Gplots also we need to plot the PCA and the other plot that we are generating in this second step. In second step, it will try to annotate the circular RNAs or read the circular RNAs, and then try to splice the circular RNAs based on the counts per million, and the data will be transformed with womb, because it is trying to identify that what reads that is identified in your sample is circular, is type of or showing the nucleotide sequences in circular format, and then
Yeah.
based on that it will try to generate the plots. Okay,
Yeah.
so this we can see in a minute. Let me just show you. Okay, so what we can do is, let me just open my. Okay, so I was doing on VirtualBox Linux, and here,
So this virtual box, is it your workstation or
no, this is like manually installed in my laptop.
so like this is also like because I'm hearing it for the first time. This is also like freely available or is it a paid one?
No, it is freely available, and we can install it in Windows when we are working in Windows and we need to run the Linux commands or tools, so we can use that.
So this is like a flavor of
Mhm.
Linux, right?
Yes,
What is
yes.
that virtual box?
VirtualBox is provided by Oracle basically.
Yeah,
So here
yeah,
is the interface where we need to generate a machine that if you can also, for example, if you are using Linux, then you can use VirtualBox to work on Windows. Okay, so it
yeah,
just provide you a platform where you can generate a new machine to run the any other type of OS
yeah.
on your machine. Okay, so
So
once we generate a new machine, then you need to give a setting option that what OS you want to use, and based on that you will be giving the access of the type of environment that you want to work.
can you tell it little slowly? Like, for example, my operating system is Windows, and
Okay.
I have seen videos of like how Ubuntu, how we can enable a subsystem for Linux in our window. So,
Yes.
this virtual box to install it for first of all, what we need to install?
Okay, so first you will be directly downloading VirtualBox from web. Like you can search, or I will share
Yeah,
you the link. Then once you download, you will get this interface itself. Okay, so now you will add open new, and then give your machine name, any name, you can use your name, and based on that, what you need ISO image. Okay, here you can see in the third text box, you need ISO image. That is for the, for example, if you want to use Linux, so for that VirtualBox website will be having the link to download ISO image. Okay, so that will be in some GBs, and then that you can download and you will provide while creating your new machine environment here. Okay, so because I am mentioning that mine is already Windows, so I need Linux. So I will be selecting Linux from here, and then it will select that what version it is. We can also modify if you are working on 32-bit, and then based on that, as I have not selected here what is the ISO image, so I need to select the ISO image. Once I am providing the name, select the ISO image that you will be uploading. It will be, you can download from website and save it in any folder. Then you need to upload the ISO image, and based on that, it will give you next step, that how much RAM and how many how much core of your CPU you are trying, you will be allowing your machine, this virtual machine, to access from your main machine. Okay, so you can give 50%, or if your disk size, storage size is 1 TB, you can give 500 GB to it. So whenever you are having the file size within your this Linux more than 500 or even around 450, it will give you warning that your disk has no space. Even the space is there in your Windows, but whatever you are allotting to your virtual box, it will be working in that limits only. Okay, so that's how it works. We can also use Linux commands in Windows using Sigwin terminal or even LTS Ubuntu that we have for that supports
But
Windows.
memory require it requires like for star it requires a high power computer, ma'am.
Yes, yes, that's why. So yeah, it requires more RAM and computation. That's why I used just the chromosome one for the reference genome. That's why, because it is not computational compatible to my machine. So for that, otherwise you need workstation to work on bigger data. But if you have less data, you want to just check with the tool that how it works, then you can always use.
Yeah.
Oh yeah, so the virtual box is good when you want to work more of most of the time in Linux, or in when you are in Windows. Okay, so here we have this first command, because let me just open the
So first step, you're doing what now?
okay, just a minute. I had to open. I am opening the script actually. Where is Yes. So we have we need, this is the folder structure when we are having, where we are having the data, and in data folder we are having the samples. So if we have the paired and reads, then two samples, otherwise we just need one sample if it is single and read. And then in FASTQ form, and then reference sec folder, where we will be having the reference genome. One file will be the FASTA file and other is annotated, that is GTF file. So
Is it necessary for the files to be .gs? Because like whenever I download from SRA toolkit, so that's always in FASTQ format. So need not to compress it, no?
no, we can work without GZ also, but don't try to download with SRA kit, because sometimes it generate corrupted files. So you can
Yeah.
download from ENA database, European Nucleotide Archive. It will provide you files in like good format for FASTQ, and also in
Using
zip.
the SRA toolkit only, right?
Sorry.
So from I have never tried with ENA, like downloading a
Okay.
file from ENA.
Yeah.
So directly you use the wget to download, no?
Yes, yes. Directly wget and search for your sample. It will provide you a link. So you can just go through. once maybe I can show you here. Because when we are downloading directly from NCBI, so it is sometimes it is giving corrupted files, and sometimes when there is a paired and reads, so one file is okay, other file is corrupted, and sometimes we have to split the files because it provide only one file for paired and reads as well. So
Yeah.
that's why it is better always to work with. So you can give your accession ID, and then you can download the link wget. For wget you will be getting the all the details. You can also cross check, but it will be giving you right only. And here we have the fastq file and the other paired and read, the second file for the paired and read. So you can use the link and download directly. You can also click, but just not to corrupt the process due to internet connection, you can use wget and download the files. Okay, so that's how it works. And let me just show you. So here is the data file where we have the sample, the fastq.gz, and then we have the reference sequence,
Yeah.
which is in okay. So I had for grcs 38 because that is the one for our sample, but just for compatibility with the machine, I use just chromosome 1 fastq and gtf file
Yeah.
for that. And during indexing, these other files were generated during indexing. So that step we perform because and the index folder, because in that also these files are there. I just copied it here. So we have the this indexing command for star, and we are running. When once we are running, then we can get the indexing log file and all the steps that is running in the indexing, because it is taking more time. That's why I generated the results already.
Yeah. Yeah.
And so this index file will be folder will be generated where all the indexing files that you are running will be listed here. Okay, so you
Yeah.
can keep it here itself, or you can copy it in your direct chromosome reference like file, so that you need don't need to change the path. Okay,
Mm.
and once after that you need to run the star command that we are having in the script. Just a minute. Am I closing the file? Okay, so text pad, it is opening in text pad. Let me just open my notepad first. Okay, here we have the command for, so this is for indexing, where we have just indexed our reference sequence, and then after indexing, we will be running the star command. So this is the star command that we are trying to run here, where we have given the junction file type and also the SAM file that we need for our sample. Okay, once we are generating, like running this star script, star command,
Mm.
then we will get the results star file. Star file. This is a star chip, which is okay. So I think results star file is here in the folder in my Linux only. Here is the result star file that we generated, and this is just for due to I don't have access. I'm using working in base Linux mode. That's why it is showing I don't have access, but I can open it. And we have the BAM file that was generated earlier. Let me just open that.
Like till alignment I have done, madam. Like I generated the BAM file. After that only I'm getting a problem.
Okay. Okay, so what after that, what you need to do, see here, we have in this is Starchip. Once you are cloning the directory, you will be getting these files. Okay,
Yeah,
and here you have the Perl script that you need to run, and also after Perl script, you have the setup shell script that you need to run. So these two files you need to run in Starship. For that, you need just the parameter file that you are having here. Okay, so
yeah,
parameter file actually is here in example files. In examples, we have Starship with
yeah,
the circular RNA, and you have the parameter. So you can just open it in your files and you can see that what all parameters you can just copy, because it is also recommended in the paper
yeah,
that you can just copy the whole script command from here and then paste it in a new file and just modify accordingly whatever is required. So now since you know that you don't have any prefix for your star results, then
yeah,
you can so yeah, so a star prefix you don't have. If you don't have any star results, then
yeah,
prefix, then you can keep it empty. If you are don't running star, then you can keep it false. And for since you are not running stars, it is mentioned not need. This is not needed if star run is false. So that's why you will either you can remove this line and or you can just add the comment a hashtag for this so that it will not run when the Perl script is running. It will not read these lines. And then you need the ID step back, that is depending on the number of files that you are having in star output. So here, since I had only one file, but in their example file, they have shown with two. So if you have two samples, then your star results will be within the file here.
yeah.
In this folder, in sample one, it's output of a star, and in sample two, it's the other output. So now you need to move two step back to come directly in the star results. So if you have the samples, number of samples, different number of samples, then you need to give two step back. And otherwise, if you have just one sample and all the results are star results are within this same folder, like junction or .sam file are in this directly in the same folder, then you can use the use
Yeah.
one, and then you can just remove these all the comments line, or you can keep if you want to have it for further reference, and then save with the same name that is given here. Okay, so let me just show you. Use all type, because if you are selecting the default text, then it will not work. So here the name is star chip circles parameter, so you can keep the same extension also. So to keep the same extension, select all types and then save. If I'm saving here, it will say that it is already there. So you need to go to your working folder and then you need to save it there. Okay.
Yes.
So for example, if we are working here, then we need to use the same file name and then save. So now our file is saved in our folder and we can use it. Then the other file type is the star output directory. So that is, we are having it here, which is star directory, star chip. And that is just for this is stardice.txt. So that is also given in example folder. Once you open your example folder, you will see both. So here is the star directory and star. These two files you need to have in your working folder to run. Okay, and this in star directory text. We have
Yes.
star out sample one. You just need to tell the script that where your star results are given. Okay, so
Yeah.
that's why we are giving id, that we are giving id step back. This is because your star directory is having the two step back in based on
Yeah.
your samples. If only one sample is there and you are giving just star out, for example, your results are in your star out folder, so you will give just this. Okay, otherwise you can just add your whole path that where, for example, let's see that if our, for example, where is results star, for example, this is my results star and I have the bem file and the junction file, and so this is not the like complete results here. First, did I delete it? Actually, I had the like this file size issues. My disk was full, that's why I moved some of the files. Okay, so if I have the for example, if I have the star results in star and here my junction and SAM file are there, so I just need to give the path of this location. For
Yeah.
example, here I will open
Ma'am,
and then yes,
ma'am,
copy
like,
this path and give it to your file so that your new star directory will be this one only. Okay,
yeah.
this is not, this is
Ma'am,
in, I'm in Linux actually. Yes, tell me.
ma'am, if you don't mind, can you keep the class tomorrow because I'm not feeling well actually now.
Okay,
I'm sorry, madam. Like,
okay, so
So, how much duration right now? It's around 1 hour. We started around 8:10, right?
yeah,
What is the time now?
it's 9:10.
Okay, so 1 hour. So it's almost the session to 9:10, right?
yes, yes.
Okay, so
Just today I'm not able
wh- how
to
much
focus.
left, Dr. Kazala? You know how
Mm,
much time it will take
I
for that?
think half an hour more will require.
Okay, what do you think? Is it possible for you to take the remaining 30 minutes tomorrow, or want to?
Yeah, that's fine. Like since it is casualty, so yeah, we'll connect tomorrow.
Okay, Sriram. So if you are not feeling okay, I
Yes.
will talk to Dr. Kazala in separate message and I will update you. Okay.
Yeah, yeah. Yes.
Okay.
Sorry, ma'am.
Okay.
No
Okay,
problem,
Sriram.
no problem. Take rest.
Yes, ma'am. Like, ma'am, I also wanted to ask, like,